Thank you. Good afternoon. Again, another beautiful summer afternoon. Today we will have two lectures. The first one is pretty long, so I plan to have an intermission in the middle of it. And then the second lecture this afternoon will be on the lysosomes, peroxisomes, and mitochondria, and that will not have an intermission. Now, for the most part, the lectures that I've been giving you are concentrating on components of the exocytic and endocytic pathways. We've covered the plasma membrane yesterday. And this afternoon, we will be considering organelles involved in the exocytic pathway and the organelles and components of the endocytic pathway. And then later on this afternoon, we will be considering peroxisomes and mitochondria. You should realize that eukaryotic cells have organelles, and this is actually quite an elaborate system of membrane-bound components. Each of these organelles has a unique composition, and they carry out distinct functions. Now, certain organelles of the cell are bounded by two membranes, whereas most of them are bound by a single membrane. Certain organelles consist of one compartment, whereas others, as we will see, have more than one compartment. Some organelles, such as peroxisomes and mitochondria, function autonomously, whereas others work in a cooperative fashion. Now, as we can see here, that there are sets of organelles that cooperate together in the exocytic pathway and in the endocytic pathway. In this lecture, the key concepts that we'll be covering are summarized here. The exocytic pathway, we'll be considering the subdomains of the endoplasmic reticulum, features of the ER at the light and EM levels. We will be covering characteristics of ribosomes and how they are targeted to the ER. Some of the key events that occurred during the synthesis of secretory and membrane proteins will be covered. Another phenomenon in the cell is positioning of proteins other than by the single sequence. And that is by localizing proteins by their messenger RNAs. We will cover that topic. We will be covering the properties and functions of the smooth ER, features of the ER export sites, the unique structure and function activities of the Golgi, different sorting and packaging activities at the trans-Golgi network, and vesicle-mediated transport within cells. Then we'll be touching upon the endocytic pathway. We will summarize the 5 major entryways of endocytosis, and then we'll be doing a comparison between clathrin-dependent receptor-mediated endocytosis and non-clathrin-dependent receptor-mediated endocytosis. Now, the endoplasmic reticulum is actually a single compartment organelle, and it has 4 distinct subdomains. These 4 distinct subdomains are the rough ER, the smooth ER, the ER export domain, and the nuclear envelope. Now, the endoplasmic reticulum is a very extensive, continuous membrane system. The EM to the left is a historic EM that dates back from 1944, and this is the electron micrograph that gave rise to the name endoplasmic reticulum. This EM was generated by studies of Keith Porter, who was one of the founding fathers of cell biology. And at that time, back in 1944, there was electron microscopes existed, but a way of producing thin sections of specimens was not available at that time. So Keith Porter had the idea of growing cells on cellophane sheets, and the fibroblasts grew and spread out on very thin sheets, and that allowed electrons to penetrate through these cells and allowed him to observe this reticular network in the cytoplasm of the fibroblasts. And this reticular network was found in the interior of the cell and did not reach to the surface membrane, which is what the arrow is pointing to here. So therefore, he referred to this network as a reticulum, and because it's within the interior of the cell, an endoplasmic reticulum. Now, quite amazingly, the micrograph that we see to the right here is a fluorescent micrograph generated from studies of 2010, and we see evidence of the same type of tubular network that Keith Porter observed back in 1944. In addition to this extensive tubular network that extends throughout the cytoplasm, there's dense areas here closer to the nucleus. And in this dense area is where we find the sheets of the endoplasmic reticulum. Now, the endoplasmic reticulum is actually a very dynamic organelle that has contacts with the cytoskeleton, namely microtubule contacts. Here in this immunofluorescence study, we see dynamics of not only the association of the ER with microtubules, but the dynamics of it moving along the course of a microtubule. The rough endoplasmic reticulum is one of the subdomains of the ER. And in this electron micrograph taken of a plasma cell, we see that the cytoplasm of the cell in this region has quite a bit of rough ER. We see that the rough ER consists of packed, parallel, flattened sacs or cisternae that are studded with ribosomes. So we see these membrane saccules, and membrane saccules are studded with these little bumps, which are the ribosomes. Now, not only are these cisternae stacked and flattened, but they can also be interconnected. So when we see this dense array of sheets of rough ER, this corresponds to what we observe by fluorescent microscopy when there's this dense labeling near the nucleus. Now, many cells have large amounts of rough ER, and these are cells that are producing a lot of protein. Plasma cells are one type of cell that is generating a lot of protein. Another type of cell that generates quite a bit of protein are neurons. And the high abundance of rough ER and ribosomes in these cells can be detected by basophilic dyes, and this is observed by clusters in the cytoplasm, which are called missile bodies. Here we're seeing more features of the rough endoplasmic reticulum by EM. And in this EM section, we're seeing different views of the rough ER. This is showing a typical view of the rough ER, where we see the cisternae, or one cisterna, and the ribosomes that are attached to the membrane. In other parts of this section, we're actually seeing grazing sections through the rough ER and the attachment of the ribosomes onto this grazing section of the rough ER. In this part of the electron micrograph, we're seeing a grazing section through the nuclear envelope region. Here is a nuclear pore, and the membrane has the associations of many ribosomes. This is what we call the polyribosome arrangement. In addition to the association of ribosomes with ER membrane and the nuclear envelope, within the cytosol we have free polyribosomes. So when we see clusters of ribosomes like this is what we call free polyribosomes. In the micrograph to the lower right is a high-resolution micrograph showing the attachment of the large subunit of the ribosome to the membrane of the ER. And then, in addition, the micrograph is showing a partition between the small and large subunit of the ribosomes. Here we're seeing a direct connection of the rough ER with the nuclear envelope. The nuclear envelope is pointed out here by the two red arrows. This arrow here is pointing to a nuclear pore. And we can see back here in the cytoplasm the rough endoplasmic reticulum, and then its attachment, direct attachment to the nuclear envelope. Now, ribosomes are, properties of ribosomes are summarized in this slide. The assembled ribosome is actually quite a tiny structure. It's only 24 nanometers in size, and it consists of two subunits. In prokaryotic ribosomes, as well as in eukaryotic ribosomes, there are two subunits. In the eukaryotic ribosome, the ribosome is larger. It has more proteins, associated proteins, and additional ribosomal RNA. The ribosomes are considered to be ancient macromolecular machines because they read the nucleotide sequence of the messenger RNA into the protein sequence using the genetic code. Now, transfer RNAs mediate the process of translating the nucleotide language of the RNA and DNA into the amino acid sequence of the protein. Now, recently, the crystal structure of the ADS eukaryotic ribosome has been resolved at 3 angstrom resolution. And that was recently generated from studies and published in 2011. And from these studies, it was shown that the ribosome consists of 4 RNA chains, which are shown in gray, and there are 79 different proteins that are shown in colored ribbons. The eukaryotic ribosome is 40% larger than the prokaryotic ribosome. Now, there's also very limited homology between the eukaryotic ribosomal proteins and the prokaryotic ribosomal proteins. And this is very important because it serves as a basis for treating bacterial infections. So with the use of antibiotics such as tetracycline, streptomycin, erythromycin, these target the prokaryotic ribosome and thereby serve as inhibitors of bacterial infections. Now, proteins are synthesized on free and bound ribosomes, and they follow different pathways. Now, initially, all proteins are synthesized in the cytoplasm. So at the beginning, early synthesis always occurs or begins in the cytosol. But then, sometimes we have ribosomes remaining in the cytoplasm, or they may associate with the rough ER. Now, those ribosomes that remain in the cytoplasm, the proteins that are synthesized on those ribosomes are destined either for the cytosol or for later incorporation inside the peroxisome, mitochondrion, or within the nucleus. This type of targeting is considered, or this type of synthesis is referred to as post-translational targeting or import. Now, on the other hand, when ribosomes are associated with the rough ER, the destination of proteins synthesized on those types of ribosomes are any of the compartments derived from the ER. And that is the ER membrane itself, the Golgi, the smooth ER, the plasma membrane, secretory vesicles, and endosomes. This type of synthesis is referred to as co-translational targeting. Now, human cells face quite a bit of a challenge to position its proteins. Our cells make more than 10,000 different proteins. In fact, some of our cells contain 1 billion proteins. So there is a problem in positioning these proteins in the cell. And as organelles were discovered, it became apparent that certain proteins are associated with certain organelles. And there had to be some kind of explanation on how these proteins became associated with specific cell organelles. Now, back in the 70s, Blobel theorized that proteins destined for the endoplasmic reticulum had a specific localization sequence. He called it a signal sequence. And indeed, his studies revealed that when proteins are translated on free ribosomes, they carried not only the protein sequence, but an additional sequence called the signal sequence. And when these proteins were analyzed on gels, they had a higher molecular weight because of the additional signal sequence. But when proteins were synthesized on microsomes that contained enzymes that cleaved off the signal peptide, when these proteins were analyzed on gels, they had a lower molecular weight. That was also seen in intact cells. This led to the discovery of the signal sequence. And the key components and events which target proteins to the ER are summarized here. Now, within the nucleus, we have the production of the messenger RNA. There's quite a bit of processing that goes on within the nucleus. There's excision of introns and rejoining of exons. And the messenger RNA is then transported into the cytoplasm. The ribosomes associate with the messenger RNA, and translation occurs. And as the polypeptide is being synthesized, the signal peptide starts to emerge out of the ribosome. This signal peptide is recognized by a signal recognition particle. This signal recognition particle binds to the signal peptide and arrests translation. Now, the signal recognition particle will bind to a docking protein in the rough ER membrane. This docking protein acts as a receptor for the signal recognition particle. Once the signal recognition particle binds to the docking protein, this brings the ribosome and the nascent chain complex close to the rough ER membrane. Then there is a release of a signal recognition particle from the signal peptide, and then translation continues. So there's a release of translational arrest. There is a continuation of translation, and the signal peptide can then enter into the channel within the rough ER membrane. Once that occurs, once there's interaction of the signal peptide within the channel protein, then the ribosome is firmly attached to the rough ER membrane. Subsequently, the signal recognition particle and the docking protein dissociate, and that involves GTP hydrolysis. There's a continuation of protein synthesis and translocation of the peptide. The signal peptide, shown in red here, is cleaved off by a signal peptidase. And then there's another enzyme that will digest the signal peptide, and that enzyme is called the signal peptide peptidase. So this summarizes some of the key events in the synthesis of a secretory protein on the rough ER membrane. Now, this slide is providing us a distinction on some of the key steps in translocation of secretory and transmembrane proteins. I'm trying to use this slide to summarize the key events, the key players that you should know: the ribosome, the signal sequence, the SRP particle and its receptor, and the channel protein. These are the key players in both the translocation of secretory proteins and transmembrane proteins. Now, this is something we just summarized in the previous slide. For transmembrane proteins, the process is actually very similar, but transmembrane proteins have a special domain, a transmembrane domain, that is recognized by the channel in the ER membrane. And the channel recognizes that transmembrane domain and undergoes a conformational change and opens up, allowing the transmembrane portion or domain to move into the bilayer of the membrane. So thereby it's attached to the membrane. So then the translation will continue until there's termination. And this allows integration of the polypeptide into the bilayer. So the significant difference between transmembrane proteins and secretory proteins is this presence of a transmembrane domain that ultimately will move through the channel of the ER membrane and be integrated into the bilayer. So there is a question on how do cytosolic proteins or cytoplasmic proteins reach destinations in the cell. I mean, there are many, many proteins in the cytoplasm, and there has to be some mechanism for their placement. And for many years, the intense focus on the signal hypothesis kind of put blinders on a lot of scientists, considering that there may be other ways to position cytosolic proteins in the cell. Recently, it has become apparent that the cell has a way of localizing cytoplasmic proteins by localizing their messenger RNAs. Here we are seeing the distribution of a specific messenger RNA for actin. and that was detected with probes to actin messenger RNA and found to be distributed to the distal region of the lamella of fibroblasts. In studies that used antisense oligonucleotides that interfered with the localization of the actin messenger RNAs led to the loss of polarized addition of actin filaments. So in the control cells here, the micrograph in A is showing the incorporation of actin in fibroblasts, and you can see that the label is polarized to the leading edge or the front of the cell. But in cells where the messenger RNA was delocalized, those cells do not have a polarized addition of actin. It's found along the whole periphery of the cell. So this tells us that for certain proteins, such as actin, that cells will first localize the messenger RNA to specific regions of the cell before even synthesis occurs. Now, in addition to protein synthesis, another important activity occurring with the ER is protein folding. Protein folding occurs as the nascent polypeptide is entering the lumen of the ER, and there are certain proteins called binding proteins or molecular chaperones that will recognize hydrophobic patches along the length of the nascent polypeptide and assist in the folding of a polypeptide. Now, in cases of cystic fibrosis, there is evidence of misfolding of the polypeptide chain, and it's essentially stuck in the ER and never reaches the cell surface. And there's been quite a bit of research interest in studying the folding of the cystic fibrosis transmembrane conductance regulator, which is a chloride ion channel protein. And it's been shown that there is a whole bunch of proteins that assist in the folding of the cystic fibrosis transmembrane conductance regulator. They exist in the lumen of the ER as well as in the cytosol. Now, there's been attempts to assist the mutant forms of the CFTR protein with pharmacological chaperones to chemically stabilize the fold and promote the insertion of the membrane protein into the cell surface. So this offers hope for treatment of cystic fibrosis. Now, the smoothie ER is a distinct ER subdomain that is directly connected to the rough ER. For the most part, we see the smooth ER as shown here. You can see that the smooth ER consists of a network of tubules that do not have any associated ribosomes. So here's our rough ER, the cysternine, and studded with the ribosomes. And then the smooth ER is devoid of ribosomes. They are not involved in synthesis as the rough ER, but have a number of other functions. Now, the smooth ER is a network of intercommunicating channels and sacs. In the liver hepatocytes, the smooth ER profiles are found near glycogen, and they are involved in the release of glucose from glucose 6-phosphate by an enzyme, glucose 6-phosphatase. Now, in skeletal muscle, the sarcoplasmic reticulum is a subdivision of the smooth ER, and it is involved in the storage of calcium ions. The smoothie R is remarkably more functionally diverse than the roughy R. Sorry. That's not part of the lecture. The smoothie R is remarkably more functionally diverse than the roughy R. And once again, note that the smoothie R is living up to its name. It doesn't have any ribosomes attached to it. Smoothie R is the site of synthesis and metabolism of fatty acids and phospholipids. It's involved in the detoxification of a wide variety of organic compounds by a group of oxidative enzymes, including cytochrome P450s. Now, the amount of smoothie R in liver cells can vary, and increased amounts are associated with a phenomenon called drug tolerance. In columnar epithelial cells, the smoothie R works to convert absorbed monoglycerides and fatty acids into triglycerides. And in endocrine cells of the gonad and the adrenal cortex, those cells have smoothie R involved in the synthesis of steroid hormones. So many times we will see the smoothie R in this tubular arrangement. But in cells, in endocrine cells, there is appearance of whorled smoothie R, as we see here. And that's due to association of proteins along the cytoplasmic membrane of the smoothie R. Now we have another domain of the ER, and that is the ER export domains. They represent a transitional ER region of the roughy R. And here is, this red circle here is focusing in on the ER export domain. So you can see that there is a blebbing off of membrane from this structure here. And ribosomes are not really attached to the ER export domain. By immunofluorescence, the ER export sites are found throughout the cytoplasm, as opposed to the localization of the Golgi complex right next to the nucleus. Now, the movement of cargo from the rough endoplasmic reticulum to the Golgi is a very important step, and it requires specific proteins and complexes called the COP2 machinery. Now, COP2 machinery, that's referring to coat protein, a specific type of coat protein called COP2. Now, at the ER export site, we have the assemblage of this cage-like structure, which consists of cytoplasmic components, such as GTPases, and heterodimeric complexes known as Sec23, Sec24, and Sec13, Sec31. And they all together assemble this coat, the COP2 coat, which functions to concentrate the cargo and to bend the ER membrane to form a transport vesicle. And this transport vesicle will butt off from the ER export sites and will move through the cytoplasm and make its way to the Golgi. So once again, this COP2 coat serves to concentrate the cargo and to bend the ER membrane to create a transport vesicle or a carrier that takes the cargo from the ER to the Golgi. This is a summary slide providing us a summary table of the various ER domains and their functions and some important associated proteins. And you need to review this table on your own study time. Now, the ER has an extensive distribution in the cytoplasm, as opposed to the Golgi, which is associated or concentrated in one particular region of the cytoplasm near the nucleus. So at this point, we're going to take a brief intermission of 5 minutes and then continue on with the discussion of the Golgi.
With the mic because I left to get a replacement battery. I heard Mary Jo told me that it built and then it popped.
Yeah.
So I told them, you can keep that on. I told them yesterday that like something's wrong with it. You shouldn't have to have it as loud as we have it, but they did not respond. Go ahead. I'm just swapping batteries. Now I am going to see what it says. It says 3 hours, but... Let's try that. It says now 7 hours instead of 3 hours, and I'm thinking that 3 hours is like really means 0. maybe. So hopefully no more noise.
Thank you. It was fun though. So with regard to the transmembrane proteins, is that specifically referring to only the endoplasmic reticulum, or how do integral proteins or transmembrane proteins get assembled in the plasma membrane itself? Do they go through a secretory process where the mRNA kind of translocates into near the plasma membrane, and then are there ribosomes there that do that? Or?
No, the plasma membrane proteins are synthesized like this. They are synthesized in the ER,
Right.
and they are inserted in the membrane, and then, as I just talked
Oh yeah,
about,
they
body
Yeah, let's go to the
time to consider the next organelle, which is the Golgi complex, which consists of stacked membrane-bound vesicles. Okay, I'll take 2 on this.
No, and it needs to be higher
Well, I'm just going to try, despite the noise. Okay, the Golgi complex consists of stacked membrane-bound vesicles and preferentially orientated tubules. Now, at the light microscope level, the Golgi complex can be seen as a lighter staining region next to the nucleus. And this is best seen in plasma cells. In both of these micrographs, we see an area that is not stained, and this corresponds to the Golgi complex region. The rest of the cytoplasm is stained because of the large amounts of rough ER present. Here's a more higher resolution micrograph of a plasma cell showing the Golgi near the nucleus, not staining like we would find with the rough ER staining in the rest of the cytoplasm. The Golgi complex position that we see in this micrograph here, where it's situated always near the nucleus, is dependent on the assembly status of the microtubules. In this micrograph to the left, we're seeing immunofluorescent detection of both the Golgi and microtubules in the cytoplasm. Now, studies have shown that the Golgi has an association with the MTOC, which is a region where we find the centrioles. When agents that depolymerize microtubules are used, such as nacabazole, the microtubules are disassembled, and the Golgi is found spread out throughout the cytoplasm. So here in the control cells with intact microtubules, the Golgi is stained near the nucleus with a ribbon-like structure. When the microtubules are depolymerized, we find a diffuse localization of the tubulin throughout the cytoplasm, and the Golgi is spread out as dots throughout the cytoplasm at the immunofluorescent level. Now, when other types of microtubule agents are used that stabilize microtubules, such as Taxol, we find that the Golgi is found at the peripheral regions of the cell. So the summary of these studies indicates that the Golgi complex, its position in the cell, is dependent on the microtubules and how they are assembled in the cell. Here's a higher mag view at the immunofluorescent level showing the Golgi in this micrograph as a yellow-orange complex or ribbon near the nucleus. We see the extensive array of microtubules. At the electron microscope level, the ribbon of Golgi is seen here. Now, the Golgi complex is organized in compact and non-compact regions. The compact region consists of membrane stacks. The non-compact region has a fenestrated appearance where there's lateral tubular interconnections of membrane. So this is an example of an isolated portion of a Golgi showing part of a compact region where there's continuous membrane and part of it showing the non-compact region, and you can see how there's a fenestration of the membrane. Here are some more features of the Golgi complex. Again, the focus here is on the positioning of the cell and the polarity of the Golgi complex. Now, you need to appreciate that in real life, there's quite a bit of activity of proteins moving through the Golgi complex. There's heavy traffic of protein and lipid. At the cis side of the Golgi, there are transfer vesicles that come from the rough ER and are found adjacent to the cis side of the Golgi. On the trans side of the Golgi, what can be seen are secretory vesicles that have dense secretory product within them. The Golgi, as we have seen in the previous fluorescent micrographs, has association with the MTOC region, and that's where we find the centrioles. I point out here that a pathology that is associated with the Golgi is known as eye cell disease. And in that disease, there's a defect in adding mannose-6-phosphate in the cis part of the Golgi to newly synthesized lysosomal enzymes. Here's some more characteristic features of the Golgi from an exocrine pancreatic cell. Here we see the Golgi, and in this case, the cisternae are dilated. Along the cis side of the Golgi, there are evidence of a number of vesicles called transfer vesicles. Further to the right here, we see an example of a transitional ER, and here is our ER export site. On the cis side, on the trans side of the Golgi, we see evidence for a contractile vacuole, and this is a vacuole that has some accumulating secretory product within the lumen. Eventually, they become larger and condense to form zymogen granules. Another important thing to notice about the Golgi region is that the density of the cytoplasm appears to be much more dense around the cisternae and just around the Golgi itself. There seems to be, you don't see much ER or ribosomes in this region of the Golgi. In this micrograph, the focus is on the vesicles associated with the Golgi. And the emphasis on two different types of vesicles. First, at the periphery of the trans stacks of the Golgi, we find dense regular coats, COP1. So these are the COP1 vesicles that are found at the periphery of the Golgi cisternae. Along the trans-Golgi network are budding vesicles that are much wider. And if you look carefully on the surface of these vesicles, the coat is spiky in appearance. And these are the clathrin-coated vesicles. Here's another example of a clathrin-coated vesicle. Here's one that's budding off. And then here is the COP1 vesicle at the periphery of the Golgi. It's important to note that there are elaborate membrane networks located at the cis and trans sides of the Golgi. And you can distinguish these membrane networks by immunolabeling studies. So the vesicular tubular clusters, which is a whole array of membrane vesicles that are found near the cis side of the Golgi, you can label these structures with a marker called ERGIC53. So that provides us a way of distinguishing the cis side of the Golgi. Now, on the other hand, if you want to distinguish the membrane networks along the trans-Golgi network, you can do immunolabeling for a trans-Golgi network marker called TGN46. Also for a mannose-6-phosphate receptor. And in this study, these smaller dots that we see here are labeling for the trans-Golgi network protein TGN46, and the larger gold dots that we see here are localizing the mannose-6-phosphate receptor. So once again, it's important to know that at the cis and trans sides of the Golgi, there are distinct membrane networks. The VTC network associated with the cis side of the Golgi, the trans-Golgi network, which is associated at the trans side of the Golgi. Now, in this slide, I've summarized some of the specific compartments of the Golgi complex. I don't want to really go through all of this. I think you can study it on your own. But basically, you should recognize that the Golgi is organized in compact and non-compact zones. The compact zone has a cis, a medial, and trans portion. The cis side of the Golgi has a vesicular tubular cluster network. The trans side of the Golgi has a trans-Golgi network. The vesicles that are directed from the ER are coated with COP2 and make their way towards the cis side of the Golgi, and they uncoat, and they either form clusters themselves or will fuse with the vesicular tubular clusters. Now, there's specific coated vesicles that assist in the membrane traffic to the Golgi from the ER, the Golgi back to the ER, and movement of protein out of the Golgi into other regions of the cell or to the cell surface. And these involve clathrin-coated vesicles, COP1, and COP2. Now, at the cis side of the Golgi, there's phosphorylation of mannose residues. This is important to note. At the medial Golgi, there is removal of the mannose residues and terminal glycosylation events. At the trans Golgi, there is sulfation and phosphorylation of amino acids. In fact, certain proteins undergo controlled proteolytic events. At the trans Golgi network, there is specific sorting and packaging of proteins. And this is assisted by the presence of specific receptors for these proteins and clathrin, which serves to form these special vesicles that transport the collected protein. Now the study of the Golgi continues, and this is an example of an advanced study reconstructing images of a Golgi within a thin section by a tomogram. And in these studies, a thin section of a Golgi is further studied by tilting a sample in one or two axes in an electron microscope, and then there's a reconstruction of the images using computer imaging. And in this study, it's possible to observe the 3D image of the Golgi. And the aqua-stained portion of the Golgi in this image here corresponds to the cis Golgi network. Here we're seeing in red portions of the trans Golgi network, and the white vesicles that we see here are the various vesicles that are associated with the Golgi. Now this sort of labeling study or reconstruction study can be coupled with immunolabeling and allows for very precise localization of enzymes in the Golgi complex. So here we're seeing the localization of manosidase enzymes to the medial cistern using a combination of thin section immunolabeling and this EM tomography, or computer-assisted imaging. So where we see labeling for the manosidase enzyme in thin section, we can find a more precise localization to the medial cistern. Now, cisternal progression maturation, as shown here in this diagram, is considered to be one of the leading explanations on how proteins are transported through the Golgi. And the evidence for this, supporting evidence from this, comes from study of large proteins like collagen fibrils. In EM studies, it's been shown that collagen fibrils will stay within a single Golgi cisterna, and that cisterna will then be gradually remodeled and will change position from the cis Golgi network to the trans Golgi network. And it's thought that even individual membrane proteins can undergo such a transport through the Golgi. Now, the case is enclosed on this model. There's some evidence that, mainly from in vitro reconstitution experiments, that specific membrane proteins may be transported through the Golgi via vesicle transport. But the leading explanation is the cisternal maturation model. Now, the movement of synthesized membrane proteins from the ER through the Golgi and onto the cell surface can be visualized in live cells. And this shows us such an experiment of the transport of a glycoprotein of the vesicular stomatitis virus. It's initially synthesized in the ER, and then it is transported into the Golgi and eventually is transported onto the cell surface. In this type of study, the G protein was expressed by a hybrid gene that encoded the G protein and a green fluorescent protein. And in addition to that, this particular viral gene was a mutant form where it was stuck in the ER at 40 degrees, but was released from the ER when the temperature was shifted down to 32 degrees. So that allowed the study of the progression of the protein from the ER, its movement into the Golgi at 40 minutes, and then by 180 minutes, it was detected on the cell surface. Now, sorting and packaging of Golgi at the trans-Golgi network is summarized here. The final phase of the exocytic pathway involves the sorting and packaging of cargo at the trans-Golgi network. And we saw from other Golgi EMs that along the trans-Golgi network there are secretory vesicles. Here we see the initial formation of such a secretory vesicle, and eventually it buds off, and then there are other stages where it concentrates within the vesicle into a denser staining structure. The diagram here provides a nice summary of the sort of events that are occurring along the trans-Golgi network. We see the budding off of cargo via clathrin-coated vesicles. There is also another type of coated vesicle called AP3 coated vesicles. There is formation of these secretory granules like this. And there are also secretory carriers that all come off the trans-Golgi network. Now, there are different types of transport vesicles that are involved in both the exocytic and endocytic pathways. And if you carefully study this diagram and break it down into different categories, it becomes quite easy to understand. Remember that I refer to the COP2 as the COP1, COP2 vesicles. COP is an acronym for coat proteins. There's two basic types of COP vesicles, COP2 and COP1. COP1 is involved in the retrograde transport from the Golgi to the ER. So these vesicles will move proteins from the Golgi back to the ER. The COP2 vesicles are involved in the forward movement of proteins from the ER to the Golgi. The clathrin-coated vesicles play a role in carrying cargo from both exocytic, that's Golgi to endosomes, and also in endocytic pathways. They are also involved in the budding off of membrane from endosomes, and for proteins to be entered into an endosomal pathway leading to lysosomes. For clathrin-coated vesicles, there are particular adapter proteins that are associated with these clathrin-coated vesicles. For those that are coming off from the trans-Golgi network, we have adapter proteins called AP1. For clathrin-coated vesicles that are coming off from the plasma membrane, the adapter proteins are AP2. And besides the clathrin-coated vesicles and the COP2 vesicles, we have formation of secretory vesicles. And they don't have a particular coat, but are either of a category or pathway that's either regulated or is constitutive. In other words, there's a continuous production or release of membrane and incorporation into the plasma membrane. Whereas a regulated secretory pathway, there's some stimulus that's required for immature secretory vesicles to move to the plasma membrane and to release their product to the outside of the cell. Now, in addition to all these different coated vesicles, you need to know that the delivery of these vesicles require another type of protein called snare proteins. And these are are particular transmembrane proteins. There's a type called a vesicle snare, called a V snare, and that will bind to a complementary target snare, or T snare, on the membrane of the acceptor compartment. And they form pairs. And once they form pairs, soon afterwards, there's fusion of this vesicle with the trans-Golgi network. Same thing here. Once this vesicle meets up with the rough ER, there you have the formation of the snare pair, and soon afterwards you have fusion of this vesicle with the rough ER. So be aware that there are special proteins called snare proteins that mediate the recognition of vesicles with their acceptor compartments inside a cell. So exocytic and endocytic pathways are the major transport pathways in the cell. And from this diagram, we can see that there's some feeding of the exocytic pathway in the endocytic pathway from the Golgi into either early endosomes or late endosomes. But before we discuss the early and late endosomes, let's first consider the process of endocytosis. There are 5 entryways for the endocytic pathway. Along the plasma membrane, cells are actively involved in the process of taking material inside the cell by vesicular transport. The 5 major pathways are phagocytosis, macropinocytosis, clathrin-mediated endocytosis, caveolin-mediated endocytosis, and pinocytosis that is not mediated either by clathrin or caveolin. Now, today in lecture we're going to focus on clathrin-mediated endocytosis, and also we'll be considering the caveoli. Now, some years ago, Michael Brown and Joseph Goldstein discovered that there's a receptor for LDL, and this receptor is missing in patients that have a genetic disease called familial hypercholesterolemia. These patients have high levels of serum cholesterol, and when cells of these patients are studied, the radioactive labeling of the LDL in this study show that there is very little binding to the cells of these patients. So these patients do not have receptors for LDL present. Now, in follow-up collaboration with Richard Anderson, he did studies with a ferritin-labeled LDL and found that the ferritin-labeled LDL was concentrated at specific sites of the plasma membrane called coated pit regions. And these are one of the first micrographs of Richard Anderson showing the labeling of and the binding of the LDL ferritin particle to a coated pit region of the plasma membrane. So here is the outer regions of the plasma membrane, and this indentation here represents the coated pit. And the ferritin tagged LDL is concentrated in this region because the receptors for LDL are concentrated in this region. Now, eventually, there is a pinching off of the membrane and the formation of a coated vesicle that enters into the cytoplasm. And you can see that the coated vesicle is in the cytoplasm, and in the interior of the vesicle, you can see the LDL ferritin particles. Well, here we're seeing some of the major components that are important for the formation of the coated pit and the coated vesicle. The important proteins are the receptors that are binding a specific ligand, and then there's an adapter complex that binds to the receptor and at the same time binds to clathrin, which is the major coat protein of the coated pit and coated vesicles. Eventually, the assemblage of the clathrin generates a network that appears to have a lattice-type pattern when viewed by quick freeze deep etch electron microscopy, and it has a very distinctive honeycomb network, either flattened or curved, depending on the extent of the blebbing or the indentation of the membrane into the cytoplasm. Now here is, once again, summary steps and the proteins involved in clathrin-mediated endocytosis. And again, the three important proteins that you should remember for the clathrin-mediated endocytosis are the adapter proteins. They serve to organize the cargo receptors and also will bind the clathrin along the cytoplasmic side of the membrane and form that network, that lattice network that we saw in the other micrograph. Dynamine is another protein that plays an important role in the formation of a coated vesicle because it serves to pinch off the membrane and subsequently release it into the cytoplasm as a coated vesicle. And once the coated vesicle is inside the cytoplasm, there is depolymerization of the clathrin coat and also release of the adaptin molecules to be recycled back again along the plasma membrane. Now, caveoli are another type of structure that is involved in a clathrin-independent endocytosis. Now, caveoli are a type of lipid raft domain, and when they are viewed by transmission electron microscope studies, they do not have a fuzzy coat associated with them like we see with the clathrin-coated vesicles. And when the caveoli are viewed by quick freeze deep etch microscopy, the cytoplasmic view of the caveoli is distinct from what we see with the clathrin-coated vesicles or coated pits. Remember, with the coated pits, we have that honeycomb lattice structure, whereas with the caveoli, there is a swirl-like structure along the cytoplasmic side of the caveoli. Now, caveoli have a number of functions. One interesting feature of caveoli is that they are involved in the endocytosis of pathogens. In fact, they are the preferred entranceway into the cell. A number of pathogens prefer to enter the cell via caveoli because once they get in the cell, they are not placed within the lysosomal pathway, so they're not subject to degradation. Other functions of caveoli include the process of transcytosis. In other words, it provides a way of moving proteins from one side of the cell to the other side of the cell in a process called transcytosis. Caveoli are also involved in receptor-mediated endocytosis, and they organize proteins and lipids in such a way to promote signal transduction in the cell. So this slide concludes our discussion of caveoli, and we'll have another short intermission, and we'll continue on with the lysosomes. diagram is emphasizing the movement of the cisternae of the Golgi, whereas this one is showing that the cisternae are fixed. The vesicles are moving. Yeah. Hi.
So I was curious, and I'm sure it's not a short answer, but you had, mentioned the folks we know in hospitalization and house residents, and we've been talking throughout the lecture about Manos. And in an earlier slide, you mentioned this, is it the eye cell disease? How does that manifest? What does that look like clinically?
Well, actually, it's going to be part of the next lecture.
Great.
So you'll get your answer.
Okay, wonderful. All right, then. Thank you.
The method that's accepted now is that the Golgi actually changes or the protein itself.
Well, it's that the protein remains within the cisterna, the Golgi, and that cisterna just moves along and is
So
modified.
it's the cisterna that is changed.
Yes.
Okay.
Right.
Thank you.
Sure.
I was wondering what causes the fuzziness in
Pardon?
I was wondering what causes the fuzziness in the image around platform.
Well, it's the presence of the code itself, the lattice forms that fuzziness.
Also, the code itself causes fuzziness.
Yes.
Is it just like
In other words, at that level, that lattice network that you're seeing when it's spread out is actually forming that spiky appearance.
that's what's coming about?
Yes.
I was wondering, do you want us to be able to identify different aspects of the landmarks that you pointed out?
Sorry.
Do you want us to be able to identify the landmarks that you pointed out on the images of like all these little initials? Yeah, that.
Well, okay, with that, here I'm giving you a way of distinguishing the cis and trans Golgi network with labeling specific proteins. So you can distinguish the cis trans side of the Golgi by these networks, membrane networks. So one would ask you, how would you distinguish the cis side of the Golgi from the trans side of the Golgi? Well, you can think of, well, there are specific membrane networks along the cis Golgi and the trans Golgi.
Now, do we need to know things like how they were able to label the TGN? Is that just kind of just for us to understand and know, but not really like examine?
No, we just need to know the protein that's associated with the trans Golgi network.
So not necessarily the specific receptor or the marker.
You should know the markers.
Should know the marker. Okay.
Sorry to interrupt. I'll let you carry on with them. I'm gonna run to my office for a sec, but just a reminder. I guess you need to wait till 3 because if you start before that, it'll cut out 2 minutes between 2:58 and 3. so a longer break. You can tell them that if you want. Or else I'll just keep coming with questions. That'll work too.
Sure.
All right. Yeah. AV is aware of this problem. They don't know how to fix it. They're going to work on it afterwards.
Oh, it's a shame.
I think, yeah, it's better, but it's not fixed.
Hi.
So here's the cis Golgi right in the trans right here. And you can see a little egg looking up.
Yes.
Now you said they get more dense, so these black things already came off the trans.
Yes. You can see there's number 1, number 2, and 3. There's different stages.
So what are these things, why are they before and after? Are they the same thing?
Well, they are probably the same thing, but they're just found on the other
They
side.
just went the wrong direction maybe? Or?
Well, they might be from another part of the complex, because remember, the Golgi is like a ribbon, and there's probably more Golgi higher up. You just have to realize that portion of the Golgi over here, and they're generating more backwards too. So that's not the only Golgi present in the cell. There might be more up here.
Thank you very much.
Once proteins are formed and they're like time for delivery, change the nutrients in that protein. What happens? Does it get delivered to, technically something given protein that was time to be delivered to the membrane. Then you change the target sequence to say maybe lysosome. Does it still get delivered to lysosome? No. Does it just get ready?
Well, each protein has its own particular, if it's destined for the lysosome, it will have a particular modification that will act it that way. Generally, you cannot change that. You know, proteins either have one type of signal to go in a certain direction, but you really cannot.
But what if there is a mutation in that signal sequence? Does that protein get degraded, or what happens to it?
I would say that it probably would get degraded.
Okay.